Hello everyone. Today I'm going to share with you a promising framework for data integration in Julia, which has the potential to simplify and standardize the methods used in microbiome research. Thanks to its general yet flexible structure, other fields of research and study designs may profit from this framework as well. First, let me introduce myself. My name is Giulio, and I study bioengineering at Rheinwald University in Germany. I'm interested in bioinformatics as a whole, and I also enjoy developing Julia packages for the analysis of microbial and biological data. Recently, I worked as a research intern at Turku University, Finland, where I helped create MicrobiomeAnalysis.jl in collaboration with the Turku Data Science Group. So the idea of an inclusive and standardized matrix-like data container for biological assays and other similar purposes has actually been around for a while in the R bioconductor ecosystem, from which this framework was also transferred to Julia. The two main components are the summarized experiment and the multi-assay experiment containers, which offer slightly different data organizations depending on the type of analysis. Here, for the sake of simplicity, I'm going to introduce only the summarized experiment object, but MicrobiomeAnalysis.jl and also its equivalent in the R language actually rely on both these and also other similar data containers. Right, now let's look into the summarized experiment object. It's made of four main components. First, one or more matrix-like assays containing abundance profiles, expression, signal frequencies, or other type of results from an experiment. This element is complemented with the corresponding metadata for the rows or features. It could be, for example, genes or proteins or taxa in the case of OTU tables and so on. And the metadata for the samples or columns, that could be maybe the information on patients, samples, or different points of a time series and so on. And then there is the general metadata about the experiment or the study as a whole. So this type of data structure gives a very solid integration framework because it includes multiple layers of an experiment in a comprehensive manner, which makes it easy to perform analysis both within and across different assays. Thanks to its general and flexible structure, this object is suitable for a number of case studies and experimental designs. And overall, the summarized experiment container has the potential to be standardized and make a few pipelines in microbiome research more reproducible. The main disadvantages of summarized experiments, in my opinion, are that it might take some learning process to handle it in a productive way, and that the needed infrastructure in Julia is still being developed. However, many applications have already been found, at least in the R bioconductor ecosystem. And now I'm going to show you how we used this container in Julia in the context of microbiome research analysis. The results from a microbiome profile or experiment generally come in the form of a matrix, where columns represent different samples or patients, and rows represent different features or taxa that were detected in a specific sample. And sometimes metadata could also come along. That's basically why we decided to build our package, MicrobiomeAnalysis.jl, on top of SummarizedExperiments.jl and its containers. Currently, we offer the features listed on this slide. Raw data can be imported from files like Excel sheets and CSV tables, but also converting it across other Julia objects is possible. And there will be more information about this last point in the next slide. For now, I'd like to focus on the modeling and visualization steps, because they often cause a bottleneck in the analytical process. And Julia, thanks to its efficiency, could actually make these two steps faster, or in some cases even feasible. A few examples of methods that could be improved are dimension reduction, like principal coordinate analysis, metric, and also non-metric multidimensional scaling, And then the analysis of time series data and simulating community dynamics are also two options that could be improved. We can perform all these different types of analysis starting from the same container, making it a shared and reproducible conceptual framework. As a side note, simulations were enabled by another package that was developed by our team called fdsolver.jl, which is meant to generate and solve systems of fractional differential equations. It was used in MicrobiomeAnalysis.jl in order to simulate complex microbial communities, as you can see in the plots on the right. This package has its own lightning talk at the JuliaCon 2022. So if you're interested, you can go and check it out. So MicrobiomeAnalysis.jl already offers a nice set of utilities, but is still under development. For the near future, there are quite a few plans and goals to be achieved. First of all, we'd like to get closer to the Eco-Julia universe. The first step is already on its way, because the community profile object, native of Microbiome.jl and more or less equivalent to a summarized experiment in terms of deliverable information, is already compatible and can be converted from and to a summarized experiment. Second, we would like to exploit the incredible efficiency of the Julia language in order to generate computationally intensive plots and develop new visualization methods for the dynamics of microbial communities. And last but not least, following the steps previously taken in the R language, we'd like to include also phylogenetic trees inside the summarized experiment object for the sake of comprehensive analysis. And these could be enabled in connection with the already existing philo.jl package. If you're interested in any of these features, please feel free to contact me on GitHub or the Julia Discourse channel, and we can work on it together. As a final note, I'd like to thank for and acknowledge the support received from Leolati and Moin Khalidi from the University of Turku, namely the Turku Data Science Group, that has mentored, consulted, and shared ideas with me. Aaron Loon for creating the packages that MicrobiomeAnalysis.jl is built upon. And finally, a huge thanks to the entire Julia community for helping me all the way throughout the journey of building a package. Thank you also for listening to my talk, and I hope you found it interesting and useful. Wish you lots of fun exploring the Julia language.